Hello and welcome everyone to the Military Women's Health Research Series. This is a joint webinar series sponsored by the Uniformed Services University's Military Women's Health Research Program and the National Institutes of Health Office of Research on Women's Health. This series will focus on crucial research topics within both women's health and military women's health. This month's topic centers on violence against women and trauma. Our webinars are held monthly on the second Thursday of each month at 3 p.m. Eastern Time. Each webinar will highlight different topics and speakers relevant to military women's health. Next month, the August 14th webinar will be focused on gynecological and maternal health. I'm Dr. Lynette Hamlin. The Uniformed Services University Military Women's Health Research Program was established in 2017, and in 2023, I was named its inaugural director. The mission of our research program is to promote, manage, and coordinate the research that will improve the health of military women through interdisciplinary collaboration and policy recommendations. Our goal through these webinars is to bring together leading experts from the Department of Defense and the National Institute of Health to share valuable insights and research findings. We're committed to highlighting critical areas of study and fostering collaboration in this important field of women's health. As we get started today, I'd like to introduce you to some of the team members providing support behind the scenes. Dr. Carolyn Bondar, Program Officer at the NIH Office of Research on Women's Health. Dr. Angela Simmons, Associate Director for Research, USU's Military Women's Health Research Program. And Ms. Christy Kennedy, Program Manager for the USU Military Women's Health Research Program. They will be moderating the Q&A later during this session. Please note that the chat has been disabled. You can enter your questions in the Q&A section on Teams. Please note that we recognize not everyone will have access to the Q&A. We do have limited time for questions at the end of the two presentations. If your questions do not get addressed, we encourage you to reach out to the presenters after the webinar. You can also contact our team member, Ms. Christy Kennedy, whose email is showing up on the screen for you for additional support. Following this webinar, we will do our best to share a copy of the presentation recording when it becomes available. We're excited to share that we are pleased to offer human-generated real-time captioning during today's event to ensure that all attendees can fully participate. You can access the stream text link through the Q&A section or by scanning the QR code that's displayed on this slide. Before I introduce our first speaker, I'd like to make two announcements. Our USU Military Women's Health Research Symposium Education Enduring activity is available for CEs and CMEs through AMSIS. The QR code can be found on this slide. Second, our program has created a forward-facing military women's health research database, which can be found by Googling Military Women's Health Research or at the address found on this slide. Now I have the distinct pleasure of introducing our first speakers. Crystal Leardman is a senior epidemiologist in the Deployment Health Research Department at the Naval Health Research Center, where she serves as the psychological program lead for the Millennial Cohort Study. For 20 years, she has conducted research on the psychological health impacts of military service, with a focus on sexual trauma, PTSD, depression, suicide, and problematic anger. Cynthia has also served as a key investigator for the Millennial Cohort Family Study and the Recruit Assessment Program. She has authored more than 70 peer-reviewed publications in leading journals, including JAMA and BMJ, and has received national recognition for her work, including the Barbara Thompson Award and the Charles Gibbs Leadership Prize. She is committed to conducting research that improves the health and well-being of service members, veterans, and their families. Joining her in this presentation is Isabel Jacobson, who is a senior research epidemiologist on the Millennial Cohort Study with nearly 20 years of experience in epidemiological research. For the last decade, she has served as the Research Portfolio Area Lead for health-related behaviors with an emphasis on topics such as substance use and sleep. She more recently took leadership of the Women's Health Portfolio with a plan to focus on topics associated with female service member readiness and veterans' well-being. Her role involves strategic planning in these areas, as well as study design and execution to provide impactful information for both the Department of Defense and Veterans Affairs leadership, stakeholders, and collaborative partners. Her work with the Millennial Cohort data has resulted in 70 peer-reviewed publications examining the health and well-being of service members and veterans. Please welcome Ms. Lierdman and Ms. Jacobson. Over to you. Thank you.
Thank you so much. We appreciate that kind introduction. I am Cynthia Lierdman, and as mentioned, I'll be joined here with Isabel. And we just thank you for joining us today. We're so excited. We're honored to be here to share some of our findings from the Millennium Cohort Program. And today we'll be talking about the impact of traumatic stressful experiences among military-connected women from the Millennium Cohort Program. Next slide. The Millennium Cohort Program is based at the Naval Health Research Center in San Diego, and we're fortunate to work with a really large, talented group of people. And just today, specifically, we wanted to acknowledge some of our co-authors of this specific project that Isabel will be presenting on today that are listed here from the Los Angeles VA, UCLA, and some of our colleagues at NHRC. Next slide. This just mentions our disclaimer, and we'd like to point out that our work is supported by the Defense Health Agency, the Defense Health Program, and the Department of Veterans Affairs, and the views expressed are those of ours, of the authors, and don't necessarily represent the government, Navy, DoD, or VA. Next slide, please. Okay, so for today's presentation, we're going to start with an overview of the program. And then I'll provide some background of the work we've done up to this point related to violence against women. And then I'll pass it over to Isabel to discuss more of the details of the Spotlight project and for our wrap up. Next slide. Okay, and next slide. The Millennium Cohort started from an act of Congress in 1999 that mandated a longitudinal study of post-deployment health among service members. And this was in response to public concern about the long-term health impacts of military service, especially among the Vietnam veterans and the veterans who served in the first Persian Gulf War. At that point, NHRC was designated as the DoD Center for Deployment Health Research, where the prospective study would be developed and launched in 2001. And while our mission to conduct high quality research on the effects of military service, which helps inform policy and program, has remained consistent over these decades, that since the inception, our program has grown, and now it has been expanded to include military spouses and adolescents, which is quite exciting. So on this next slide, you'll see that the three studies here. So the Millennium Cohort study was launched in 2001. That's the study of the service member. And we have about 260,000 participants in that service member study. Then in 2011, the Millennium Cohort family study was launched, and that is following military spouses over time. And then most recently, the Millennium Cohort study of adolescents, which we refer to as SOAR, was launched, and that's following military children, specifically adolescents, who are linked to the Millennium Cohort participants. Together, these three studies comprise the Millennium Cohort Program, which you'll hear us referring to. Next slide. This figure shows the enrollment, the survey cycles over time of the Millennium Cohort Program. So you can see, for example, the service members, which are here indicated in red. Our first panel was enrolled between 2001 and 2003, and then we've continually surveyed them approximately every 3 to 5 years. You can see in green a similar format for our military spouses, the Millennium Cohort Family Study, and a similar format for our adolescents, which are depicted in blue here. Next slide, please. So just to give a little bit more overview of the Millennium Cohort Study specifically, it is the largest and longest running cohort of U.S. service members. There are currently over 260,000 participants enrolled, and the cohort includes service members. It's quite broad, from all service branches, all components, including Reserve and National Guardsmen. And one unique aspect of our cohort is that we continue to follow the participants even after they leave service. So this really helps us to get at that transition point, leaving the military and then becoming a veteran. In addition, not only do we collect a broad array of survey data, including mental, physical health, along with exposures and occupational experiences, but we also link to a number of external databases, such as medical records, mortality databases. So the integration really creates this comprehensive, multifaceted data set that's uniquely positioned to conduct these longitudinal analyses. Next slide, please. So while the data from Millennium Cohort Study is used to examine a broad array of topics, today we'll specifically be focusing on the research related to violence against women. So the next few minutes, I'll just give a brief background of some of the previous work we've done in this area. And this research has been conducted by a large group of our Millennium Cohort researchers and numerous collaborators over the decades. Next slide, please. Some of our initial work examined the impact of sexual violence. So we found that women who experienced sexual harassment or sexual assault had a higher likelihood of PTSD, depression, and multiple somatic symptoms. Further, when we examined to see how these relationships differed by various factors, we found that while the associations remained fairly stable across most demographics and military factors, the associations varied by other types of trauma. For example, on the figure to the right, you can see a simple linear pattern of increase in the odds of PTSD by childhood trauma among those with no sexual trauma. However, this pattern is more nuanced among those who've experienced sexual harassment or sexual assault, indicating a kind of ceiling effect, especially among those who've experienced more than one type of trauma. Next slide, please. A few other studies have examined other impacts of sexual violence. For example, we found that women who experienced sexual assault were 73% more likely to resume unhealthy alcohol use, and they were twice as likely to have difficulties at work compared to those who did not experience sexual assault. In addition, women who were sexually harassed were more likely to be demoted while serving in the military. Next slide, please. Examined contextual factors of sexual assault associated with PTSD using data from our 2019-2021 survey cycle of the sexual assault survivors. And what we found after adjusting for demographic and time since most recent sexual assault is that individuals assaulted while serving the military had an increased risk of PTSD compared to those who were assaulted prior to joining the service. And results further indicated PTSD risk increased if the perpetrator outranked the victim or was a spouse of the victim or a significant other. Further, incidents that occurred at military installations were also associated with significantly higher risk of PTSD. However, perception of support, including support from the military, served as significant protective factors. against PTSD. Next slide. Other research has examined aspects of domestic abuse. Two studies, which are shown here on the left in the light purple, linked family advocacy program reports of physical and emotional domestic abuse perpetration with service member survey data. Of the multiple conditions examined, PTSD and alcohol dependence presented unique risk for future domestic abuse perpetration. Trauma-specific PTSD symptoms appeared to increase the risk of perpetration through negative effect and specifically anger and irritability. And this relationship held true both for men and women. On the right is a study that is underway. It's using Millennium Cohort family data to explore service member mental health in relation to spouse reports of domestic abuse. And this is using a screener on our surveys. And what you can see is problematic anger of the service member had the strongest and most direct association with domestic violence compared to other mental health conditions. And further, the findings suggest a stronger likelihood that pre-existing PTSD may exacerbate problematic anger and indirectly increase domestic abuse risk. Next slide. We've also explored the relationship between sexual trauma and sexual health. As we know, sexual health is an important component of well-being. And what we found is women who were experiencing sexual assault were more likely to have worse sexual health. And this relationship was partially mediated through PTSD. The findings suggest that treatment options and insurance coverage for sexual health problems may need to be expanded among our DoD service members and our veterans. Next slide. Lastly, more recently, we have started to examine sexual assault among military spouses. Again, this is using data from the Millennium Cohort Family Study. And what we found is since entering the military community, 8% of spouses experience sexual assault. And these prevalences were higher among military spouses who were serving in the military or previously served in the military. And the sexual offenders included friends, relatives, spouses, significant others, and other military personnel. And now I'd like to turn the presentation over to Isabel, who will discuss the findings from the Post-Trauma Insomnia and Mental Health Project.
Thank you, Cynthia, and good afternoon, everyone. I'm going to shift gears a bit and go into more detail on one specific study that we felt really spoke to the theme of this particular webinar about violence against women. So this paper was about the contribution of post-trauma insomnia to depression and post-traumatic stress disorder in women service members. Next slide, please. So the main aim of this particular study was to determine whether insomnia played a role in the association between recent traumatic experiences and mental health outcomes among female service members and veterans. For this study, we were able to include more than 26,000 female service members and veterans across all service components and branches, again, regardless of separation status. Next slide, please. This slide shows the methods for the study, including the time frame. So on the left in the red box, we have our time one, which was measured at our 2011 to 2013 survey cycle. And at this time point, we measured the recent traumatic experiences. So these were experiences that were reported by the women to have occurred within the last 3 years of taking their survey. And these included combat, sexual assault, and sexual harassment. We also measured probable clinical insomnia at this time point using a cutoff score of 15 or greater from the Insomnia Severity Index. And so the reason that we looked at the 2011 survey as our time one for this study was that this was the first time that any of the Millennium Cohort surveys had included the Insomnia Severity Index. So that was our time one. And then 3 to 5 years later, at time two, which was collected at our 2014 to 2016 survey cycle, that's when we measured our outcomes of probable PTSD, which we measured using the PTSD patient checklist, and then probable depression, which we measured using the patient health questionnaire. And also just a note about the terminology of probable when we're talking about the insomnia, PTSD, and depression. The reason we're using that term is because these are self-reported survey items rather than a physician diagnosed condition. So just want to be on the same page with everybody about that. And then in terms of our methods, we used mediation models and a path analysis to look at the relationship between the trauma experiences through insomnia to the mental health outcomes. So here we're jumping in looking at our characteristics of the study population at time 1. And what we can see is that at the bottom table, we can see that 53% of the women had separated by time 1. So when we talk about our military characteristics, this is meaning for those who are still serving, that represents their current status in the military, while for those who had separated, that represents their last known status in the military prior to separation. So in terms of our sample, the women were 37 and a half years of age on average. About 70% were non-Hispanic white, 74% were enlisted, 55% were in the reserve or guard, almost 70% had higher than a high school education, 54% were married. And as we can see from our pie chart on the right, the majority of the women participants were either serving in the Army or the Air Force. Next slide. Okay, here we are looking at the prevalence of our exposures, our mediator, and our outcomes. So on the left, we have our exposures, which were again measured at time 1, and we can see that recent combat experience was the most prevalent of the exposures at 14.4%. Recent sexual assault was reported by just over 2% of women, and recent sexual harassment was reported by 5.5% of women. In terms of our mediator of insomnia, that was quite prevalent at 18.6%. And then regarding our outcomes for probable PTSD, 10.5% of women screened positive, while just over 8% of women screened positive for probable depression. Next slide, please. Okay, so here we are jumping into the fun part of this, which was the bread and butter of the analyses for this study from the mediation model. But there are a lot of numbers and arrows here, but we're going to walk through them step by step to make it a bit more digestible. So on this slide, I'd like you to focus on the red arrows and those numbers, and that is the first part of the research question here, which is, are the recent traumatic experiences associated with probable insomnia? And what you can see if you're able to read those numbers, these are odds ratios and 95% confidence intervals, that yes, indeed, there was a positive and statistically significant relationship between each of these traumatic experiences and probable insomnia. Next slide. Okay, so now we're focused on the second part of our research question, which was, is probable insomnia then in turn associated with our outcomes? And what we can see here, again, focusing on the red, is that indeed there was an association. It was significant, and we can see that women who screen positive for probable insomnia were more than twice as likely to then screen positive for PTSD or depression. Next slide. Okay, so then on this set of slides in the diagram, we're looking at direct effects that remain after we account for that mediating factor of insomnia. So what we see here, again, if looking at the red arrows, is the direct effects that remain for recent combat experience to both of our outcomes. And what we can see is, again, that there is a significant relationship. Next slide. Okay, here we have, again, direct effects that remain between recent sexual assault and probable PTSD or depression. And we can see here that women who reported recent sexual assault are twice as likely as those who did not report sexual assault to screen positive for PTSD or depression. Next slide, please. And then this is our last set where we're looking at the remaining direct effects. And we can also see here that, again, we have a significant relationship between recent sexual harassment and probable PTSD and depression. Next slide. Okay, so taking this all together, again, it is a lot to sort of digest, but really what the take-home message from this all is that the traumatic experiences, all three individually were significantly associated with probable insomnia, and probable insomnia in turn was then associated with both of our outcomes. So probable insomnia was a partial mediator, and that helped explain those effects. Next slide, please. Okay, so what does this mean though in a real world context? Because those are a bit of complex analyses and sometimes a bit hard to digest. So how do we apply that to an actual real world setting, and why does it matter? So the reason this matters is because this study, the findings from this study, helped us to be able to recommend that we should be prioritizing screening for sleep problems. So if we're not screening for insomnia, then we're likely not diagnosing or treating it. So if a woman presents to a provider and has experienced a traumatic event, then there is an opportunity there to screen and potentially treat for insomnia, and a missed opportunity if not done. So the idea is that if we can intervene at that earlier point on insomnia, then this might actually mitigate the development of the post-traumatic mental health symptoms down the road, as we saw from our analyses that would potentially develop, because those post-traumatic mental health symptoms are often more complex and quite a bit more costly to treat than versus treating insomnia. And then secondly, we can also recommend prioritizing women-focused research. I think everybody here probably attending the webinar today agrees how important this is, but we really know that there are just, unique needs of women service members, and they're really understudied, and there's so much more to learn. So anything that we can do as a community to help funnel dollars towards women's health research is really needed in order to ultimately improve resilience, lethality, and ultimately retention among our women service members, so that they can be not only the best version of themselves while in service and on the battlefield, but also after they separate from service and as they are with their family members as well. So that's what we're, ultimately we would love to be able to contribute so much more in this area. Next slide. So that wraps up that particular talk. That paper is published and available. Our contact information is here. We're happy to, engage with anyone who would like to, either later during the Q&A or after the talk. Thank you so much.
Thank you so much. And you can stop sharing, Christy, and we'll let Dr. Duncan get her slides up. And Dr. Tigno, do you want to proceed with introducing Dr. Duncan?
Sorry, took a while for the unmute button. Well, thank you very much for that wonderful presentation. My name is Senya Tigno. I am the Associate Director for Careers of the Office of Research on Women's Health, which we call ORWH. The ORWH was established in 1990, so we are celebrating our 35th anniversary this year. And this office serves as a focal point for health research at the National Institutes of Health, which is focused on women. And so we work across the NIH and all the 27 institutes and centers to advance the understanding of biological and social factors influencing women's health and disease. But we also support individuals in biomedical careers and stimulate research to improve the overall health of the population. So biomedical researchers, particularly those involved in women's health, remain part of our mission. And this afternoon, I am very pleased to introduce Dr. Laramie Duncan, who is an assistant professor of psychiatry and behavioral sciences at Stanford University. And she has a PhD in neuroscience and clinical psychology from the University of Colorado. She also continued to have clinical training at the Harvard Medical School and did postdoctoral work in statistical genetics at the Broad Institute of MIT and Harvard. She has led genome-wide association studies, or GWAS, for major international consortia. So she has done pioneering work integrating GWAS data with single-cell and single-nucleus RNA sequencing and data from the human brain. She's identified specific brain cell types associated with PTSD, depression, schizophrenia, and related disorders. Having a combination of computational skills and training in the foundations of clinical interventions in neuroscience, Dr. Duncan has a track record of executing high-impact projects, and accordingly, her work has been cited over 29,000 times in major journals, including Nature, Science, and Cell. So without further ado, please welcome Dr. Laramie Duncan. Thank you.
Okay, so hello everyone. It's really an honor to be here. Thank you so much to the organizers for making this webinar, and it's really a pleasure to present alongside the other scientists that we're speaking today. I'm going to shift gears a little bit from the previous talk and talk about what, as I was putting this presentation together, I realized is really an NIH success story. And beyond that, this is really truly more broadly a success story for science and for community. And you'll see why I'm saying that in a moment. As far as we know, these findings apply to women and to men. And at the end of the talk, if there's time, I can explain why we think that to be the case. I'll be talking about massive data sets and statistical analyses, but I want to make sure that this talk is accessible. So I want to start by getting us all on the same page, and I'll give the terminology that's necessary to explain how we do this work and what the importance of it is. So we all know when we think about the human brain and the human experience that there is a tremendous amount of complexity. We also know that the untreated burden of mental health problems is massive and it's growing. The previous presentations really spoke to this. We know that this is especially true for our military service members, and that there's much that needs to be done to make better use of the treatments that we have and also to develop better treatments. In terms of PTSD and other psychiatric disorders, environmental factors like trauma and social support and broader societal factors will always be relevant, and so that's one area of research and work that will always be a priority. The other major factor that's also very important is genetics. It's now well established that genetic factors influence all psychiatric disorders from a moderate to a large degree. And the first point that I want to make today is that these genetic influences are useful if we're trying to understand the causes of PTSD and other psychiatric disorders. So why do I say that? Why do I say that these genetic influences are useful? Well, despite the fact that the human genome is vast, it encompasses 3.2 billion base pairs in each one of our cells, we have the technologies now, thanks to decades of NIH research and investment from other groups around the world. We can systematically study the DNA systematically and inexpensively. For less than $100, we can get essentially all of the genetic variation in a person's DNA or RNA. The type of study that I want to first mention is referred to as a genome-wide association study. These are straightforward studies, but their results are really important. And so I'll explain to you how they work. We know that our human DNA is packaged into these chromosomes, these 23 packages of DNA, which are in almost all of our cells. When we conduct these genome-wide association studies these days, we're typically examining about 10 million genetic variations scattered across these chromosomes. So this is a very in-depth study of very minute things within our bodies. And the study design, like I mentioned, it's very straightforward. We collect a group of people that has a PTSD diagnosis and attempt to find people that are good matches in terms of being controls that don't have that diagnosis. And then we make comparisons, looking at these nucleotides that we represent with these letters, A's, T's, G's, and C's, to try to find any locations in the genome where there is a systematic difference between cases and controls. And as I'll mention again, we do this about 10 million times for each genome-wide association study. Here I want to point to the community aspect of this work. I'm showing just two people here. Patrick Sullivan of the UNC started the Psychiatric Genomics Consortium, which is a group of about 2,000 scientists around the world that have led these genomic studies of psychiatric disorders. And Kerstin Koenen, who I've had the pleasure of working with for about a decade, started the PTSD group within the PGC. You may know her work from other aspects of PTSD research, and so many others around the world. So the way that this work proceeds, and I led the first analysis and writing of the first paper from the PGC PTSD group. The way it works is that we line up the genetic variations across these chromosomes along the x-axis. These are chromosomes 1, 2, 3, 4, and the y-axis denotes statistical significance. And so what we're looking for is any locations in the genome that we call loci, where we have genetic hits. Anything surpassing this threshold here means that this is a difference that surpasses an incredibly high threshold for statistical significance that is an international standard. So in 2018, when we published this paper, and this is work that had taken about 5 years to complete, we had about 20,000 people in this combined study. And the question was, how many genetic hits would we get? It turns out that the answer was 0. So we had, even with a sample size of over 20,000 people, 0 genetic variants that surpassed this threshold. I'm really proud of the fact that we published this as a null result for this portion of the work, because it was true that we just didn't have any true genetic hits for PTSD at this point in time, and neither had any other study. There were group members within the group that wanted to publish subgroup analyses as significant, but Kerstin, of course, supported us in not doing that. Because we already knew at the time, even in 2018, even well before that, we knew that we might need more people in the study to actually detect the genetic hits that do contribute to PTSD. And so we continued the work, this time with the major paper led by Caroline Nievergelt's group. In 2024, actually, this paper was eventually published in Nature Genetics, reporting 95 genetic hits contributing to PTSD. Over 1 million participants contributed to this study. So it took a truly massive US and international effort involving also the Million Veterans Program and lots of military and civilian studies to eventually identify these genetic hits contributing to PTSD. And it's not only these 95 genetic hits, but there are many more that we know that we will be able to discover eventually. So is this cause for celebration? Well, certainly it is. But we also know, and have known for well over a decade, that this was actually only going to be the first step. Because the whole point in doing a genome-wide association study is to scour the genome and find novel clues about the biology of these disorders. So this result that we have right here about PTSD, with about 95 loci for PTSD, we were at this point in research for schizophrenia about a decade ago, when it became absolutely clear that determining the biological significance of these loci was a major challenge in and of itself. And really, again, that shouldn't have been a surprise. That's why we conducted these genome-wide association studies. So I could talk today about all of the different ways that people have tried to go about determining the biological significance of these loci. And this has been my passion and my interest from the start. There are a number of approaches for doing this, which you may have heard of some of these. I could talk about any of these. I know I'm familiar with these methods, but the reality is that each of these has major limitations. It's not that the statistical methods are incorrect, it's that in large part, they are just vastly underpowered. In contrast to that, there's one approach, one way of combining GWAS results with data about cell types in the human brain that actually gives us a systematic, well-powered approach to interpreting psychiatric GWAS results. And so that's what I'm so excited to share with you now. It's this way of making sense of the psychiatric GWAS results to get a cellular understanding of brain phenotypes. Our first paper in this space was published earlier this year, just in January of 2025. So I'm oversimplifying here just a little bit, but in broad terms, it's correct that we now have a good sense of genes that are associated with psychiatric disorders. Now what we need to know is, what are the cell types in the human brain? So I'll make the argument that not knowing the parts of the human brain, not knowing the cell types, makes us, puts us at a profound disadvantage. And this is the technology, the single nucleus or single cell RNA sequencing. This is the technology that allowed us to actually know the cell types in the human brain. For the sake of time, I can't go into too much detail, but this is the motivating idea, and this is what makes these studies so successful. So despite the fact that in our body we have the same genome, no one would mistake a lung for the brain. These tissues are very different from one another. They're different because different genes are used in different combinations and amounts in different tissues in the body. The same is true for different cells in the brain. And that was what was assayed in this 2023 science paper, which is from Sten Linnarsson's group at the Karolinska Institute in Sweden, where they sampled from human brains of donors that contributed their brains to science. They sampled 105 regions from across the human brain and over 3 million individual cells from those parts of the brain. And what they were able to do was to figure out how many cell types are there in the human brain. And you could ask leading neuroscientists this question, even up until now and very recently, and they might give you numbers of a couple dozen cell types in the human brain. What this study and the similar studies have shown is that unequivocally, there are at least hundreds of cell types in the human brain, likely a couple of thousands of cell types in the human brain. For the sake of time, I can't show you the beauty of these studies, but there's literally hundreds of studies now that can show the fact that the cell types that result from these analyses are truly real. They're not merely clustering artifacts. What it shows really beautifully, this study that was published in 2023, was that there are at least 461 cell types in the human brain, or at least a good draft of these 461 cell types, as I'm depicting here. So what I'm showing is that there are these non-neuronal cell types here in grayscale, and the rainbow colors represent neuronal cell types, the information processing cell types in the human brain. And I'm going to keep showing this rainbow bar representing these human brain 461 cell types. And the benefit of this data set, this major discovery by the Linnarsson group and other groups around the world that have pursued these same approaches, is that we can see the cell types in the human brain. We now have a draft parts list of the human brain. And what we did in our lab was we combined these two critical data resources, asking which cells use the genes that are important for particular psychiatric disorders, such as PTSD, schizophrenia, and depression. This is the way that we combine the results based on very robust, well-established methods with some extensions that make our method a little bit more powerful and accurate. And we put it together. So this is a very sophisticated audience. Many of you may be thinking, of course you can construct an algorithm, but does it really work? So that's the right question to be asking. That's the question that we were asking ourselves and that other groups have asked. It turns out that our method does identify the true positives, as they are known for many different complex brain phenotypes. This article by Dr. Ian Fife, who I don't know, but he's the editor of Nature Reviews Neurology. He's very familiar with all the research about Alzheimer's disease and multiple sclerosis, which are neurological conditions. And he writes here that the results that are from those for those disorders validate this approach. And that's why we're able to delve into the results for schizophrenia and other complex brain phenotypes here. So for the sake of time, I'll just skip past the point, but the point being here that our approach can identify very different types of cells, such as immune cells for multiple sclerosis, different types of neurons for sleep duration and alcohol per week. Using the same approach, we get different results for different phenotypes, and they appear to be correct. So our first results and work in this space is published in Nature Neuroscience, Duncan et al., 2025. These are the schizophrenia results, which of course I can't go in too much detail about this. And these are the PTSD results. And I'll show you a couple of highlights about the PTSD results. So now stepping back and looking at in a very broad picture, how these results look for different psychiatric and related phenotypes, such as sleep, which is related to insomnia, of course. Looking across psychiatric disorders, we always see that there are cell types in the cortex, that is this outer gray matter part of the brain, and in the amygdala that are associated with these conditions. But perhaps as many would have expected for PTSD, which is a fear-linked disorder, the amygdala is the structure of the brain most closely linked to fear. We see that amygdala cell types are most prominent for PTSD. And it's thought that the process of fear conditioning explains various aspects of the etiology and the symptoms of PTSD. And these are exactly the brain regions that we would expect if we were looking at a fear conditioning paradigm of PTSD. So the amygdala, the hippocampus, cortex, and thalamus. Again, comparing across different phenotypes for these mood disorders, we also see basal forebrain cell types. For sleep, we see the cortex and amygdala cell types, but also cell types deeper into the brain, including the pons and medulla, where you would expect these cell types. So this is at a 10,000-foot level, the results from our studies, but I just wanted to give you a sense of some of the overarching conclusions from this work. And reiterating again here that for PTSD, as contrasted with all of the other brain phenotypes that we've looked at so far, we see a greater fraction of amygdala cell types associated with PTSD than any other condition. So in many ways, our first paper in this space was dedicated to showing that this approach really does work, was dedicated to showing that the method is valid. And so it might seem like we've just recapitulated what was known before. But the point that we're making is that this is a totally new, far more detailed path forward. I think we can sort of think about it this way. If we have a very old map of North America and we're trying to find someone's home with an address, we might get roughly into the right location, but it's very unlikely that we would find what we are looking for. But now we have something like Google Maps, where we can zoom in to incredible detail, and we're very likely to find what we're looking for. So just putting this another way, I think that as long ago as 20 years ago, probably much longer, it was clear that one of the issues in psychiatry was that biology is incredibly complex. We don't know really where to look. Even very, very considerably, if we're thinking about just 20 brain regions, only 100 cell types, only 10,000 genes, this works out to 20 million possibilities. Where in the world should we look? Where should we focus our attention to try to understand what are the biological elements that give rise to these conditions and make some people more vulnerable to others? What we've done with this work is we've been able to reduce the search space in a principled manner with well-powered data sets that are both systematic and genome-wide and brain-wide. So to just ground this in what we're talking about for PTSD, going from sort of looking across the whole brain, we now know that we should look at the amygdala. That was known before, but it turns out that there are probably 200 different cell types in the amygdala. Now we know what cell types to focus on, and even within those cell types, by virtue of the way that these data sets are generated, we know what genes and what receptors are used by these cell types. And so now we have novel drug targets and novel ways of thinking about how we would target these cell types in a precise manner to try to treat these conditions. So needless to say, we're very, very excited about this work. There's so many future extensions of what we can do with this work. We want to expand phenotypes. We want to look at developmental cell type data sets, many other things. So we have this clear path forward. I'm very thankful and grateful to have an amazing team and collaborators ready to assist us. We're, running forward with this work. This is what keeps us running into the work, running into the office in the morning and working on this after our kids go to sleep at night. We're so excited. I will be honest too that we've already had funds cut, and it's hard with this uncertain funding landscape. We'll figure out how to do this, but I can't give this talk without acknowledging the juxtaposition between all of the work that's gone up to getting us to this place and the real success that has occurred thanks to sustained investment by NIH and other research groups over the decades, and the incredible hard work of so many people to get to this point where we finally have a data-driven, systematic approach to understanding the biological basis of these conditions and a way forward to developing treatments in an intentional way, as opposed to the luck that developed the medication treatments that we had previously for psychiatric disorders. So we're just really trying to figure out how to continue this work forward and not lose momentum. I'll close by saying what we found. So we've been able to find precise cell types associated with PTSD, schizophrenia, and other psychiatric disorders. What's really important is that these cell types were discovered with an entirely data-driven, systematic approach. And this used only well-powered data sets. In the paper, we were able to show and demonstrate that we do have adequate power for these analyses, because that's always important. And again, these are genome-wide, brain-wide data sets that we used from humans. What this gives us is a new, robust understanding of the cellular basis of psychiatric disorders, as well as a roadmap forward. We want to finally show the pathology of these conditions. So certainly there's a biological instantiation of the genetic basis of these disorders, but it's been subtle, so we haven't been able to show it very clearly, but we think that now that we know where to look and exactly what to label, we should be able to do that. And there's lots of other opportunities for intentionally developing and repurposing drugs, potentially even being able to match patients to treatments. So I'll close by just saying thank you to the amazing team and to the organizers of this event for having the opportunity to speak. And thank you all so much.
Thank you so much to both our speakers. You can stop sharing your slides and we will move into the Q&A. I think Angela is going to get us started.
Thank you to our presenters for doing such a great job, and of course to you guys for attending. I will start with one question that was asked in the chat to our first set of presenters, Ms. Linderman and Ms. Jacobson. And that question was regarding insomnia. And it says insomnia is also a symptom of the late reproductive stage from ages approximately 35 to 45 and perimenopause from 45 to 55. Did you control for life stage in your study?
So we controlled for age, which, yes, thank you for, that's a great question, because I don't think I mentioned what was controlled for in the mediation models. It was all of the variables. I did show on one slide, like the time one characteristics. We had age, service branch, some of those military characteristics, as well as whether or not they were separated from the military, but not life stage, so to speak, in terms of like, premenopause, perimenopause, things like that. So yeah, that was, the adjustment was mostly age, I would say, for that. But great point and good question.
Okay, we're waiting. We got a few. If you can please place your questions in the chat. If Christy, we have a question. If you can put the link for the CEUs in the chat again, that would be great. We have one question, Dr. Duncan. How can someone join your research team?
Oh, what a nice question. Feel free to email, but yeah, thanks so much.
Okay, we'll give it a couple more minutes.
There are hands up.
Oh, I did not see. Yes, we do have hands up. Thank you. Okay, so Autumn Rodriguez, I'll go ahead and give you the mic. Should be able to ask your question now.
That was an accident. Sorry about that.
Okay. Then we'll move on to Harinder. I'm sorry if that is not the correct pronunciation. You should be able to ask your question now.
Me?
Yep.
Thank you. Just wanted to know that chat is still disabled. But I definitely have some questions. Thank you for this. It's been very insightful. And And I just want to say that as women, we are definitely more likely to encounter the need for healthcare services through our lifetime, and that many providers can be dismissive of any symptoms and particularly mental health or mood concerns. Women deserve a better delivery of care and support in situations of violence and trauma, not only medically, but socially and legally. These are life-altering, debilitating experiences that have the potential to burden a woman until the end of their time. What are your proposed strategies to overcome this?
So I don't know if either of our speakers want to speak to that.
Sure, I'll try to say, I mean, kind of one of the points, I mean, not sure how well it was made, but of the first talk at the end was kind of just that. It's, you know, women may go in and seek care, but they're a lot of times, you know, it is dismissed and it's like, oh, you're having a sleep problem, here's a drug, go away, you know. And so I think part of what we want to do is really bring awareness to these issues with women and try to, you know, so I think with awareness becomes knowledge and becomes power, and empowering women to even speak up for themselves when they do go in to see a provider. I mean, often that time is short, but if they can advocate for themselves, that's, you know, one way, but also increasing the awareness among the providers that, hey, these needs are specific. Like, we can't dismiss these women. We need to treat them with respect and help them. And I think that message is, you know, starting to percolate. But again, it takes time to change those types, you know, societal norms. But I think that, you know, we're all here today as a research community, and we're trying to help shift that and move that needle over.
Thank you so much. And we are at time, so I am going to ask Christy to bring up our closing slide, and we'll have Dr. Tigno close us out. Thank you so much to our speakers and everyone for attending. It's been wonderful. We have such a wide array of participants from all geographic areas and sectors, so thank you so much.
Yes, thank you very much, Carolyn, and thank you, Dr. Hamlin and team. So we here at ORWH are really truly pleased to be able to present this series of webinars to you. Next month, we have another exciting issue on gynecological and maternal health. We hope you can attend. And every month thereafter until December, we have planned out our series. So please do keep coming back for issues that may be of interest to you or to your family. Thank you very much all for attending. See you next month.